Hi everyone, I'm Naomy Aviles Diaz, and today I'm going to talk a little bit about my project here as a Sigler Fellow regarding microcystis interactions with associated bacteria and control on bloom characteristics within specifically Western Lake Erie. My mentors were Anders Kildal, Sara Rivera, and Lauren Hart from the University of Michigan, and Greg Dick from Sigler. So a little bit about me and why I got here and how I learned about this program. So I am from the University of Puerto Rico, where I'm pursuing a master's in biology. And here I study very different environment. I study solar salterns, which are about 30% salt. So it's 10 times higher than the content of marine environments. So very salty, very different from water from the lakes. And the ecology there is also fairly different. We study mostly the diversity of the organisms in those salterns, but also the ecological relationships with other organisms. And studying those, I started taking a course in limnology and continental bodies of water, where I learned a lot about the Great Lakes, and I found the opportunity here. And then from there, I kind of wanted to apply ecological studies that I have been doing in the past about interactions between organisms, but in a more lake setting. So I think that we've heard a lot about how important harmful algal blooms are within the Great Lakes. So these are basically caused by the proliferation of algae or cyanobacteria in the surface of aquatic, but also marine environments. And they're casually called HABs. Specifically within Western Lake Erie, we have the concern of the CHABs, or cyanobacterial harmful algal blooms. These are disruptive to the ecosystem because they can create a big accumulation of mass that affects light penetration, oxygenation, and various chemical and physical factors of the lake, but also more specifically because of the production and release of secondary metabolites. We often hear about the toxins and the water quality of the region. Between these secondary metabolites, a particular toxin that interests us a lot of the time is Microsystem, that is often produced by Microcystis. So Microcystis as a species, as a genus, has an extensive and diverse biosynthetic pathways. They are able to produce these very complex molecules that are very interesting, but also harmful to the ecology. And some of these metabolites likely also modulate the interactions with associated bacteria. And just a little bit of a catch, like these metabolites are indeed like toxins too. So the geomicrobiology lab, which I was working with this summer in the University of Michigan, they have a collection of different strains of Microcystis that produce different amount of toxins, but also have different numbers and diversity of other bacterial strains that are related to them. And here are a couple of examples of those different Microcystis strains. But my interest was more focused on those bacteria that associate with Microcystis. Why? Because we know that Microcystis exist as a colonial form, which means that they produce the EPS, or extracellular polymeric substance, that allows other bacteria to attach to it and form interactions, and the typical ecological interactions. So you would find beneficial, neutral, and then negative interactions. An example of beneficial interactions that Microcystis could have with other bacteria is receiving vitamins such as vitamin B12 that they cannot regularly produce. Neutral interactions, where they're simply existing together in this microenvironment, but also negative interactions, where we can have bacteria maybe degrading some of the toxins that Microcystis is producing, or maybe even degrading that polymeric substance and allowing for Microcystis to not aggregate as easily, which is good for us because then it could mean that Microcystis is not accumulated to the same density at the top layer of the lake, but for the Microcystis ecology themselves, it's a negative effect on them. So studying the ecology of Microcystis and how these how the bloom formation occurs can allow scientists to generate better strategies on how to control blooms within the Great Lakes. And to start my project, one of the first steps that we took was to start isolating this bacteria from the field, specifically during bloom season, which started during June. I was able to go to a lake and sample 2 times this summer in June and July. We would filter water from 4 different stations using 100 micron filters, and we would also collect whole water samples. Then we would go to the lab and realize a couple of serial dilutions where we would dilute the water contents through series of growth media, selecting for specifically what we know as heterotrophs. Heterotrophs are bacteria that consume organic compounds and organic carbon from outside sources, so they themselves do not produce it, which is a difference between Microcystis that produces its own carbon sources. But we still used a media that was slightly oligotrophic, so it didn't have rich nutrients to assimilate the environment in the lake, but a little bit so that we would select for those bacteria that are associated to photosynthetic organisms such as Microcystis. And once we started to see growth, we would isolate those colonies with morphologies that were significantly different because we wanted to cover a variety of different organisms. And once we started collecting these colonies, we started purifying and making sure that they were pure. And we would go through them through microscopy and validify that these were indeed scenic cultures, where we only saw one type of morphology of cell, at least for what we could tell with microscopy. And then we would pick a colony from there and proceed to go through 16S RNA colony PCR, where we basically choose this gene known as 16S. Is a taxonomical barcode that allows it to identify different taxonomical groups of bacteria and possibly archaea. And here we would make many copies of this gene so we can eventually validify that we did indeed have a bacteria, and eventually proceed to sequence it through nanopore long read sequencing, which was a very interesting methodology of sequencing because we would get results pretty quickly. So about in a day we would get results. That's a pretty interesting sequencing method. So out of all the different strains that I was able to isolate this summer during the fellowship, I was able to identify a significant diverse cohort of isolates. So the geomicrobiology lab has a heterotroph collection, just as the Microcystis collection, because we've seen how important these organisms can be. And all those that have a dot or a circle on them were isolated this summer by my work during the fellowship period. And if you could pay attention to the bottom part, there's a lot of diversity that was seen for the first time, at least in isolates, during this year. So there's entire groups that were found during the isolation of organisms this year, such as the Bacillus species and Ixobacterium right here, and also a very interesting one, Deinococcus. So to resume that big collection of organisms, I made just a smaller, more concise tree of the different genera that we were able to isolate this summer. And 3 of the ones that interested me the most were Deinococcus, Rosynas, and Porphyrbacter. Porphyrbacter was the one strain that we were able to isolate more abundantly within the samples, but there could be bias to why. These are very pretty colonies, have very distinctive colorations, and we were trying to find a specific morphology based on data that we have read in the literature. But it has also been seen in important interactions with Microcystis themselves. Then Deinococcus is known to be an organism that outside of this environment has been proven to be resistant to high levels of radiation. So that is an important and maybe possibly interesting interaction that they could potentially have. But most importantly, and I think the highlight of my summer here was isolating a strain of Rosynas. So Rosynas has been previously found through sequencing the genetic content of Microcystis colonies themselves, so taking everything that is inside the little colony of Microcystis and all the associated bacteria, sequencing all of the different components in there, and kind of finding a relationship of frequency of the different strains that are found there. And Rosynas was often found directly relating to the Microcystis colonies, but it had not been previously isolated from the lake by the geomicrobiology lab, at least. And there's also a couple of other uncultured families here, like the Microcystis family, that we were able to find strains that belong to these families and could possibly also have a direct relationship with Microcystis. So a second part of my project was then seeing how heterotrophs or associated bacteria grow in response to the exposure of supplementation with metabolites coming from Microcystis strains from spent media specifically. So these are metabolites that we extract from cultures where Microcystis was present, and all the residue from the extracellular components that they liberate, we are able then to supplement it to different bacteria and see how they utilize them. Specifically, the strains of Microcystis that were used to choose these metabolites from were those that had a significant difference in the different production of metabolites. So Microcystis, as mentioned before, is able to produce diverse biosynthetic gene clusters or possess diverse biosynthetic gene clusters, such as the production of different toxins, such as microcystin, cyanopeptolins, and anabionopeptins. So we try to choose those strains within the culture collection that presented a variety of different pathways. So in green, we would have the ones that possess the biosynthetic pathway for that specific metabolite. In gray is that they didn't have a pathway for that metabolite specifically. And then in yellow, they had a partial route for the biosynthesis of that specific metabolite. So once we chose those 5 strains of metabolites, we would then proceed to also choose strains of associated bacteria that were pretty different from each other, because we want to see how different groups of bacteria react to the exposure of different metabolites from different strains of Microcystis. So we would take these different heterotrophs or associated bacteria and we would culture them with the different types of metabolites, and we will set them up in a couple round of these experiments. It's not just one set of experiments, it was a couple variations of these experiments where we would have a couple of controls just to see how they naturally grow and if the metabolite was indeed having an effect on the growth of these strains. But then we will also have a variation of the concentration of metabolite. So we would have 1/10 of the metabolites found in the spent media, or that Microcystis was secreting to the media, and then we will have the same amount of the concentration that was found in the spent media, and then 10 times the concentration of those metabolites. Once we have those set up, we can then generate growth curves and see specifically how the growth of this different bacteria occurs within the presence of Microcystis metabolites. So for all of the mentioned before associated bacteria, one specifically that really caught my attention and got very interesting results was a strain of Erythrobacter species, in which we would see that for at least 2 of those metabolites coming from the strains of Microcystis, LE1941 and LE1955A, there was a increment of the standard growth rate of these organisms. So they were growing at a higher rate when exposed to higher concentrations of metabolites. So this could probably tell us that in some way they're utilizing those metabolites for their growth, because the media that they were naturally growing didn't have a lot of nutrients themselves. So this increment in growth could be related in some way to the exposure of these new metabolites. The metabolites for these two very drastic increments come from strains that one of them does contain the microcystin pathway. So it could be of interest in future projects to possibly study if they're in some way degrading microcystis or doing something interacting with that particle. And one of them also didn't have the cluster. So what else within those metabolites are they using? Could be a potential question for future experiments. And the overall association between growth of the bacteria and the metabolites, I highlighted a couple of different relationships that I found very interesting, especially between these two strains of heterotrophs. There was an increment of growth rate when they were exposed to the same amount or the same concentration of metabolite that was known to be in the original Microcystis media. And then as soon as we increased that, they depleted their growth rate. So there could be a relationship between concentrations of metabolites and their growth when exposed to them. So conclusions on this project is that there is a number of uncultured heterotrophs that can change the ecology within Microcystis colonies and therefore bloom formation. So these organisms can have a relationship in how fast or how strongly Microcystis can aggregate, and possibly how it also produces toxins, which is a particular interest in the Great Lakes. And Microcystis interactions with other microorganisms could be regulated by secondary metabolites, so they might be producing metabolites that either recruit other organisms or doesn't allow organisms to partake in interactions with them. And the isolation of these microorganisms within Western Lake Erie will allow scientists to conduct laboratory experiments and identify appropriate strategies for bloom control, maybe identify genes that can have a use for the field. Some future questions. After finding some of those strains, I would be really excited to see how their genome looks like, so possibly sequencing those genomes and identifying specific genes that could allow us to have a better understanding of how they are interacting with Microcystis, and then even go into a lab and actually putting to test those theories. And are these strains isolated, are abundant within Western Lake Erie blooms? This could also be done by correlating the strains with metagenomic data, so the data of the entire DNA that is found within Lake Erie. And how specific are these interactions, and do they co-evolve? Because it's been shown that Rosynas specifically has a relationship of co-evolving with Microcystis, at least in other lakes and in other strains of Microcystis around the world. So seeing that within the Great Lakes would be pretty interesting. And then through growth experiments, do my particular strains isolated this year have a positive or negative impact in Microcystis colony formation? And just for an extra fun thing that could be found within those genes, is there any bioremediation prospects? Because usually these heterotrophs have the ability to decompose very interesting materials, and just looking over those genomes to see if there is the identification of those genes. So some acknowledgements, and I really benefited from sharing a lot of time with different groups of people this summer. So I was able to use a lot of help from Esme Coupier, a past member from the Geomicrobiology Lab, who worked on this project before me and was able to generate a bunch of different data before me so I could compare it and also build from her project. Ian Fierro was another fellow from the Geomicrobiology Lab, and he was working directly with Microcystis, and I was able to isolate associated bacteria from his plates. And from Sigler, I am very thankful for all the help and the guidance that Mary Ogdahl, Margaret, and Aubrey offered us through this summer. All the lunch and learns, everything that you guys gave us was really useful. And everyone at Sigler that was able to support us. And the entire Geomicrobiology Lab for welcoming me and guiding me through this summer, specifically my mentors, Anders Kildahl, who was there like every week keeping up to date with my project and everything that was going on, Sara Rivera, Lauren Hart, and Greg Dick.
Naomi. Great job. Questions for Naomi?
Okay, so you mentioned that your work in Puerto Rico is very different from your work that you conducted this summer. So I'm kind of interested to hear, are there any lessons learned or like comparative situations that you can bring back to your work there, and maybe other things that we can learn from your experience in that ecosystem as well?
I feel like in lab work, I will bring a lot of different experiences and methodologies back home that I had to a lot of the times troubleshoot by myself. I'm the only graduate student in my lab there. So a lot of the times I had to figure things by myself. And I feel like having the experience to work in a lab that has a different set of methods to do things would allow me to really troubleshoot things in a very efficient way when I go back. But also I feel like the dynamics within the lab were an example or a role model lab, in my opinion, for how things should be worked within labs, and the dynamics of how information was shared and how everyone just was really connected with each other's work was really special.